G'day Randall. This is going to be a quick interview about the up-and-coming conference in New York, the GF2045 conference in June 15th and 16th. So first of all, let's just start by saying, well, what is GF2045?
Okay. Hi, Adam. Yeah. So as you already said, this is a congress that's happening on June 15th and 16th. It's called the Global Future 2045 International Congress, and we're doing it at the Lincoln Center in New York City. It has a subtitle called Towards a New Strategy for Human Evolution. And this is because we're focusing on a number of developments regarding to the body and the brain of humans or our species, and we're trying to put it in the context of social change and our culture and civilization and where we should go with that. What sort of threats are there? What sort of challenges are we going to face? And how do we deal with that by allowing ourselves to move forward in some fashion? This is where the evolution part comes in. So it brings together cutting edge science that's involved with a roadmap towards extending ourselves, our minds, and it tries to combine that with the concerns that people have everywhere by bringing in also some representative thought leaders who can speak to us about their view of what matters in civilization.
Excellent. So, and the website is gf2045.com, just so people know how to
Ah,
get
yes,
that.
right. Yeah.
Yeah. So,
hmm
well, what is unique about the conference? That you'd think people would be able to take away from this?
Well, mostly the types of congresses or conferences you see are aimed at talking about either just social issues, such as when you have a large UN conference or something of that nature, or an economics conference, something of that kind. Or, on the other hand, you've got scientific conferences, which are very technical, very scientific. They just address projects and scientific interests, like the Society for Neuroscience annual meeting, for example. And then you've also got conferences that are indeed aimed at futuristic topics. They bring in futuristic speakers and speculate or predict. I'm thinking here about H plus and transhumanist conferences like the Singularity Summit. But the Congress that we're putting possible solutions, it's bringing together issues of civilization and culture and looking at it through the lens of what can we do in technology and science, all on the cutting edge here, and to bring it together to produce new and incredible solutions that move us forward. So the Congress aims to present the problems, but also to present solutions with at least one option of a complete roadmap. And this roadmap, at least, is aiming at brains and how to safeguard or extend and improve those. So my specific job at the Congress is to make sure that that roadmap and all the projects that tie into it are presented clearly by the speakers and by myself. I think that it's a really good time to have a Congress like this because it so happens that this is the moment, this is the time when there is a convergence in the brain sciences from many different angles. There was a period between 2008 and 2012 where we saw an incredible advance in the field known as connectomics. That's the field in which we analyze the connections in the brain, the architecture of the brain, and that culminated in a number of big projects. And for example, we have in Europe projects that are based on the Blue Brain project, where they're making models of an entire column of neurons in the rat, and this is now turned into the Human Brain Project. And here in the United States, just recently, we had the Brain Initiative that was announced by Obama and the White House. This was a really fascinating, exciting time. I couldn't sleep the night before that came out because it's looking at the other side of that coin, at the activity in the brain and making brain activity maps. We've seen increases in new neurotechnologies such as optogenetics, neuroprosthetics, synthetic that's being applied in the brain areas. All of this is coming together right now, and we're also showcasing all of that at the Congress and how it fits together. So it's very central. The Congress is right now very central to this entire convergence.
Well, you did mention some really interesting. You mentioned that there's the neuroscience and theory of mind track. Now, there's going to be some really interesting speakers like Theodore Berger, Boyden, Ken Hayworth, and yourself. Do you want to talk about some of the speakers that are going to be talking in that particular track?
Yeah, sure. Yeah. So you mentioned a few of them. I'm just going to give you a whole list of people that are either talking about something in the area of theory of mind or neuroengineering. We've got Marvin Minsky there, who's going to be talking about intelligence and his prediction that uploading is really just a matter of of time. Sorry. By uploading, I mean being able to transfer minds into other types of machines, like computing machines that are not based on our biological components. Ben Goertzel, Dr. Ben Goertzel, is going to be talking about artificial intelligence as well as uploading, I believe, because he's a proponent of both of those. Anders Sandberg from Oxford is going to be addressing whole brain emulation in the context of social and ethical considerations. Ray Kurzweil, who was recently hired by Google and is well known for his futuristic presentations as well as his work in artificial intelligence, is going to be talking about his ideas around brain building. He recently put out a new book on that.
Hmm.
Theodore Berger is going to present his neuroprosthetic work. He was recently hailed by the MIT Technology Reviews as having produced one of the 10 breakthrough technologies of our time.
Yeah, that's
And Ed Boyden, well known for his work in optogenetics at MIT, and also a great builder of neural interfaces and other tools to work with the brain, is going to present exactly that, new tools and how they can tackle the problem of recording and stimulating at high resolution and large scale. George Church, who is well known for his work previously in the area of well, I guess the Human Brain Project, the Human Genome Project in genetics, is now moving a lot of his work into the area of how do we tackle problems in the brain, and he's looking at using synthetic biology tools. This is his specialty to aid in neural interfacing, data acquisition, and brain building. One of those is a fascinating project that I hope to talk about a little bit with you as well, the so-called molecular ticker tape.
mm,
Then myself, Randal Koene, I'm going to present the big picture aspects of this. So, what are all these brain aspects of what the Congress is presenting? How do the speakers' projects connect? And what does the roadmap look like? What are the challenges that we're going to come across, and what can we expect in the near future? Dunin Markovsky is going to talk about his approach to understanding the systems of the brain, and I think he's basically trying to approach the same topics that the Blue Brain Project is addressing, but he's using a different method there. And Ken Hayworth is going to talk about technology development for the preservation and readout of the brain's connectomes, so the brain's connections and architecture. And finally, we also have Hammeroff and Penrose, as well as Panoff, who all three of them are going to be presenting some alternative theories about possible mechanisms at fundamental levels of brain function.
okay, that sounds interesting. Yeah, I have hooked up an interview with Ken Hayworth, so I'm looking forward to that. So yeah, well, that's a star-studded cast, definitely with Marvin Minsky, especially at the and Ray Kurzweil, I guess. But yeah, Marvin Minsky is one of the
Mm-hmm.
the early pioneers of AI. So there we go. So it does really seem like there's a lot of interesting approaches to understanding the brain that are popping up like mushrooms all over the place. You mentioned a couple. Some of them I don't think a whole lot of people are familiar with. Like people may have heard of optogenetics, but that's still maybe almost fringe. I'm not sure. On the fringe of the consciousness of public awareness. But molecular ticker tape. Now we've spoken about this before in a previous interview, but that sounds really exciting. Do you think that that's
Yeah.
got a lot of potential in the future?
So the molecular ticker tape is the first example, I would say, of attempting to use our synthetic biology tools to produce devices that can either acquire data or stimulate or produce some effect, not just in one place, but all throughout an organism and in a very targeted manner. We're going to see a lot more of that in the future. But this first example, the molecular ticker tape, is meant to be a way to record activity that's going on, record changes in cells and specifically neurons, and to be able to do so at a very large scale but high resolution. I think it's a fascinating approach that I don't think can get into all the details right here, but I definitely think that's going to be an example of the sort of thing we're going to see in the next 5 years or so. Yeah.
Wow. Yeah. So I mean, there's a lot of technologies that have popped up. Now you mentioned Brainbow before. And that's a really interesting one.
Sorry, I'm having trouble hearing you right
Oh,
now.
sorry, you mentioned brainbow before. Brainbow,
Rainbow.
yeah,
Yes.
sort of the way that they can color separate neurons.
Hmm.
That is quite different from the early approaches by using silver filaments or some sort of silver
Yeah.
compound. And that's been very interesting for scientists to be able to determine
Mhm.
which neurons connecting to what other neurons, but also it got some attention in the public eye because it looks beautiful.
Yeah, I think both of those things are really important. For the scientists, I think the most important thing about rainbow and similar techniques is that previously you could only apply one stain at a time. If you wanted to use more stains in the same tissue, you'd have to wash out the previous stain, and there were only so many times you could do that. Plus, you would never know if you could get a really good stain and see much of what was in there, even with one stain. With Brainbow, you get an idea of just how dense everything is in the neural tissue. You can really see a number of different types of neurons, different cells, glial cells at the same time, see how they're all connected, see how they're all kind of twisted around one another and parallel and going in various directions. This is something that's really good scientists to look at. It's great for them to see all of it together in one circuit. It's also useful for everyone else to get an idea of, oh, this is really what our brains look like. It's not just a piece of meat or something. It's not some kind of uniform tissue. There's a whole lot going on here. It's a circuit. There are wires everywhere. There are components everywhere. It looks like a very complicated computer panel that you just ripped open and you see all the wires spilling out of it. So these beautiful color pictures, just having that out there in the media, it really inspires people to look at the brain as a complex mechanism.
So yeah, GF2045 is covering a lot of these wonderful technologies, and well, hats off to Ed Boyden and well the team who have been pursuing the optogenetics. Now, how do you think that optogenetics seems? Where do you think that that's going to go? How would it actually help us understand what's going on in the brain, and also maybe
Hmm.
engineer sort of ways to
Yeah,
help improve the brain function with like
yeah.
improving like people's quality of life, for instance.
So optogenetics is a method of stimulating neurons. It's a way in which you can prime neurons by genetically altering them from birth. But then in the future, there will also be ways of using gene therapy to apply it in existing adult brains to change the neurons so that they become more sensitive to light. All neurons are already sensitive to light, just like all matter is sensitive to various types of signals. But in this particular application, you can make the neurons sensitive to different colors or different frequencies of light so that, for example, one frequency will turn them on and another will turn them off. You've got green light and blue light doing opposite things. And if you can do that, and you can do that by targeting specific types of neurons, then you know, for example, that when you are delivering that light stimulus and you're stimulating just a specific set of neurons in an area of the brain, and then you look at an MRI image and see what's going on, or you're recording with electrodes, you have an idea which neurons are producing the effect that you're seeing in your recording. So you can get a more specific recording by having a more specific stimulation. And in this sense, optogenetics is an incredibly powerful tool both for stimulation and for recording, so and for analysis. It's a way of modifying the activity that's going on in the brain by stimulating, by turning on and off, but it's also a way of reading out more information, understanding what you're looking at. And I think that's just the first example of tools like that. I know, for example, that these same labs that were involved in developing optogenetics are now also looking at ways of creating new biological tools to record from the brain instead of just stimulating the brain. So they're going to go a long ways to producing more of these types of results. Yeah.
That's amazing. Yeah. So, well, what about the Henry Markham's Blue Brain project? You mentioned some of the differences between the European Human Brain Project. And you said there were some similarities, or that was actually an extension of Henry Markham's work. Is that correct? And if so, how is it an extension?
I'm not entirely sure that I heard your entire question, but if I can
Okay,
interpret it correctly, it, one thing that's important to understand is that the Human Brain Project is a project that is putting together work in a number of different labs and a number of different universities, institutions throughout Europe to understand more about the brain from the point of view that was already pioneered by the Blue Brain Project.
sure.
The Blue Brain Project took samples from many different animals, many rodents, many rats, and looked at those to get at a lot of data. For example, looking at the architecture within a column of the neocortex of the rat, and to see which type of cell connects with which other type of cell, and at what distance, and with how many synapses, generally speaking, and which ones of those channels, those connections are going vertical, or which ones are going horizontal, all that kind of architectural information, but also a lot of dynamic information, such as which type of synaptic channels do these cells have and how do they communicate, what sorts of neurotransmitters are involved there. So they did a lot of analysis on neural tissue, and they used that to produce a stochastic representation of what the circuitry in a column is like. And using this stochastic representation, they built a model of a typical column. So this is about typical brain modeling. You're trying to make models that represent on average what you would expect to see. And then if you run them through functional simulations, you can see the type of rhythmic activity or the type of propagation of activity that you would also recognize by looking at an MRI or recording with microelectrodes. It's different than, for example, trying to build a neuroprosthetic for someone. It's different than trying to make a system or a representation of a neural circuit that is specific to any one individual or any one specific animal. And in that sense, the Brain Initiative, which is this initiative that was just recently announced in the United States, is quite different because it wants to look explicitly at creating brain activity maps, at making tools that can collect a large amount of data from a very specific piece of neural tissue, from a very specific brain, and represent that. And this is in sort of a, it's sort of the next step after having built a lot of connectomics tools, being able to acquire the architecture from a specific piece of neural tissue. So this is all about representing what's going on in that brain, in your brain, in one particular brain. And I think those two things, so the Human Brain Project and the Brain Initiative, are complementary in this way because the Human Brain Project provides a lot of information, very fundamental, basic information about, first of all, how do we acquire kind of data? How do we look in there and find out what the neurotransmitters are, what the architecture is, what sort of tools do you need for that? How does it usually end up looking? How is it supposed to work in general?
Hmm.
And then in the Brain Initiative, we're going to get tools to look at specific brains and be able to do things like make neural interfaces and neuroprostheses.
Okay, so you did cut out there just for a second, but just to clarify, um, what are the similarities between the Whole Brain Project and the Obama Brain Initiative or the Brain Initiative in the USA again?
Okay, so the similarities are that both of the projects are really approaching the brain from a neuroinformatics perspective, so that it is considered a very large, complex system, and you're looking at it at the level that computational neuroscience deals with these things. The difference between the two projects is that in the Human Brain Project, it's mostly about building typical or I guess, an abstract model representing what a piece of neural circuitry would look like in a type of animal. Whereas in the Brain Initiative, it's more about recording data, recording architectural data, recording dynamic data, and recording that in a very specific piece of neural tissue, in a very specific animal or person, acquiring that data and using it to, for example, make neural interfaces or make a prosthetic to replace a piece of the brain.
Right. So it's good they're not exactly covering the same ground, but they seem to
No,
be
they're
covering
complementary. Yeah.
different ground, but complementary enough so that they can sort of use each other's data in the future if needed. I imagine. Yes. Is that would that be reasonable to say?
Yeah, I think that's quite reasonable. I think they're very complementary indeed.
So if there's anything missing, do you think from both of these initiatives that isn't being done at the moment?
Yeah. Whether or not something is missing there, I guess we can't really tell yet because we don't know which parts of the Brain Initiative are actually going to be implemented and which parts are just being proposed at this time. It hasn't gone through Congress yet. There hasn't been an okay. So we don't really know, first of all. That's one thing where there might be something missing. The other is that we're already hearing voices that are arguing about where specifically these funds should go. So again, there's a matter of what is it really going to be applied to, which projects are going to go ahead. And of course, there are still parts of this whole puzzle that are going to require attention and that haven't yet really been an emphasis, not neither in the Human Brain Project nor in the Brain Initiative. And one that I think of, for example, is the problem of taking all of this data that you acquire and being able to map it to a representation that works, so a model, and that in fact produces the type of activity and the same activity that you've previously seen in your original sample. That's totally non-trivial. If you have a sea of data, picking through that and coming up with the right models and the right parameters isn't easy at all. Right now, some of the early proof of principle work that's been done, specifically in retina by Brigman et al. And Bach et al., they worked in visual cortex, actually, Bach et al., has been along those lines, but fairly simple because it was dealing with input and output that was known. We knew that the retina is receiving light, for example, and we know very much about those cells that are there, and we know kind of what they're supposed to do. And so it's very easy to interpret what's going on. In other parts of the brain, it's not that easy. So making that mapping between data and model, that's going to be a big, difficult step. In fact, even with the work that Brigman et al. Did, most of the work so far was done by having students sit down and manually classify what was in the images they were looking at, which is not something you can do if you're going to be tackling a huge volume of neural tissue. So there are going to have to be some developments in the recognition of what's in these images, of doing automatic model building, automatic parameter extraction, that sort of thing.
Yeah, right. So, I mean, with machine learning techniques, do you think we have the
Yeah.
current techniques
Yeah.
to be able to do this image recognition?
I think
Or
that the current techniques for image recognition are good enough. It's more a matter of teaching them or building the systems in which they know what to look for.
yeah,
So they have to take that expert knowledge that the student has, that the students who were classifying these images had, and put that into a machine learning algorithm.
yeah. So we need to really train these machine learning algorithms. But I mean, we need to train across large data sets. The data sets that we have now, are they good enough? Could we use our current data sets to train
Oh, I think
the
some of them are. Some of the same data sets that the labs of Winfried Dank et al., the ones that they collected and used for these publications that came out in 2011, they're quite good. They're definitely usable for work on automatic techniques for object recognition in slides of neural tissue. It's just that that work has only just begun. There are some projects out there to work on it, maybe not as many as we'd like to see. There certainly isn't an entire initiative that's focused on this representation challenge. Um, but yeah, there's definitely some data that you could use to start working on it. Yeah.
This morning I was looking at the World Bank's sort of statistics on how much GDP was actually focused on research and development and science, right? And Israel's
Hmm.
ahead. I mean, it's been progressively going up. It looks as though there is a scale, and it seems as though more and more money is being put into this. But Israel's doing pretty well. They're, I think, about 4.5, 4.7% of their GDP is focused on R&D. America have got 2.7%. Um, and
Yeah.
and and this is and well, Russia's got about 1.03 or something. I'm not sure. Australia's somewhere in between. But the top five countries seems to be like Israel, Sweden, Denmark, Korea, Japan, and the USA is up there, but it's still about seventh on the list, which is a
Hmm.
little bit concerning. They have a lot of money, right? There's
Yeah.
it'd be nice to see some of the larger countries with huge amounts of money pushing a lot of
Right. Yeah.
finance towards research and development, especially neuroscience. I mean,
Hmm.
this is something that philosophers have been like wondering and pondering and really excited about since we've been recording things, right? Since we've been recording history, it's so evident in in the old text, in the philosophy, and in some of the like even some of the religions. Just a self understanding. It seems to be we have that chance to do it now, right? A very exciting time to be alive. So
Yeah,
it
yeah, that's
wouldn't be
true. It's a
great if
very
we could.
exciting time. Yeah, it's a very exciting time because the resources are there in principle if we want to put them there, and because we've reached a point where where we know what to do with the resources. We know which topics need to be addressed. So I think when you see a country like Israel put a fairly large amount of money into neuroscience, it's very clear that it has an effect because Israel is known in the neuroscience community as one of the cutting edge advanced areas where neuroscience is happening and where a lot of the great researchers are coming from right now, at least percentage wise as a per capita amount. Of course, the United States is so large that even if their GDP percentage being put into this is somewhat smaller, it's still a very large amount. It's a major powerhouse in the area of neuroscience. But I think one of the things that needs to happen, and I hope that the Congress is going to help with that, is that there needs to be a connect between this science and the stage that we're at and why it's so exciting that there are so many things happening there and why this matters to people. Why does this matter to the politicians? Why does this matter to our economy? Why does it matter to society? How is it going to help us? How does it actually connect to the future of our civilization rather than being just some pet hobby that someone is carrying out?
Yeah, yeah. Well, I think it's a good thing that I think the GF2045 initiative has actually written a letter to the UK about this, right? There's it's written to Kim Moon. What I forget. I think you better talk about that.
Yeah.
Yeah, yeah, yeah.
Yeah, I
So,
mean,
what's the gist of that letter that they've sent?
yeah, you can look at that from a number of different ways. Writing a letter to the UN is perhaps not the most traditional step that people working in the sciences would do, but for someone who's interested in it from a civilization perspective, from a perspective of development and evolution and where are we going with this, what should our priorities be, it does make sense to address this to groups and authorities who normally think about where should we go and what are the important priorities for our nations and our leaders to be thinking about. Whether or not that quickly has an effect, the effect that we'd like to see, whether or not people really take it seriously and realize it, we'll just have to see. Maybe we need to push this in a number of different ways to get the important message across. But I think, yeah, it was a good thing to try. We'll see where it goes.
Yeah, yeah, true. But it's interesting. I mean, like, there are a lot of there are a lot of ways to fund these scientific initiatives. A lot of scientists wouldn't say no to more funding to help them perform more experiments and get their research done a lot quicker. There's roadblocks there, scientists don't want to spend all their life writing fund or grant proposals either. They actually want to get work done. So yeah, I really hope that there's a lot more funding. Now there seems to be a lot more private funders funding these sorts of things. On the news recently, there's been the talk of.
I, I'm having trouble hearing you, actually.
Oh, okay. Is this? Can you hear me now? All right. Yeah, I was just suggesting that there seems. I was just suggesting that there seems to be.
I can't hear you well right.
All right, all right. It's probably just a temporary glitch in in
Yeah.
bandwidth land. Can you hear me now?
I can hear you. Yep.
Okay, all right.
Yep.
So just quickly, what I was just saying is there seems to be an increase in the amount of funding from a government level. Scientists would
Hmm.
like to have more funding. They would like to spend less time writing grant proposals and actually
Yeah.
have more time to get things done.
Yeah.
But then again, from another coming from another direction, there seems to be a lot more private funders.
Hmm.
This Yuri, the Facebook, the guy who
Right.
sort of
Yeah.
found
I mean,
Facebook, and
Yuri Milner is one example who's getting into it, but Paul Allen is, of course, the prime example of someone who is putting money into neuroscience through the Allen Institute.
Yes.
And it's a very interesting model because although an individual philanthropist may not be able to put in the kinds of funds overall that a government can put in, a philanthropist can pick a specific area, such as, for example, mapping out the brain or something of that nature, and can really drive that one area forward because just focusing on something essential like that, focusing on the tools, focusing on acquiring that data, you can do a lot with the sort of funding that a philanthropic organization can put in. So seeing what the Paul Allen Institute has done, but also some of the work that's happening at the Salk Institute and at some of the other institutes that are more privately funded or that have some other sources of funding, it's very interesting to see where that's going these years. And I would definitely suggest that for someone who wants to have an effect on the future, for someone who is a sponsor, a supporter of something they think is important, they should also look into the brain sciences and into this work that's happening in the area of understanding our brains, the area of building brains, in the area of augmenting brains and neural interfaces, because it's something that is going to pay off big in not just in the sense of producing some kind of financial reward, but in the sense that it is going to make a huge difference to what we can understand about ourselves, how we can relate to each other, and how we can move forward and make our whole society more flexible and more able to deal with various challenges coming up.
Yeah, that's interesting. It reminds me of an interview I did with Margaret Bowden, and also another one I did with Aaron Sloman, actually. And some of the topics they brought up were really interesting, and it's all about asking the right sorts of questions. And if we just did a blanket research initiative on the brain, we might
Yeah.
not get there as quickly as if we were asking the right questions. So maybe our philanthropists can actually get some more insight into, well, what are the right questions? Actually, what do you think are the right questions to ask when trying to understand, well, in neuroscience now, what's being asked? What's some examples of some good questions that have been asked, and what's some examples of questions and experiments that you would do
Mm.
to to further the neurosciences that
I
aren't
think
being
that
focused on this?
most of our big questions in neuroscience, psychology, cognitive science, philosophy are addressing things that you can really only understand once you can look at the interaction of different brain regions, and once you can look at the specifics of the mechanism underlying what's going on in the brain. So when we talk, for example, about things like perception, about consciousness, about awareness, about executive function and decision making and all this other stuff, all these big topics are addressing things where the brain is making use of many different regions at the same time, and those regions are doing very specific things. So when we have assumptions about what's going on, or when we're trying to debate whether things work one way or another, what you really want is you want to be able to look at the exact activity explicitly, mechanisms that are going on in these circuits, but you want to be able to do that across different areas of the brain, looking at them in synchrony, how they interact. So this is all a matter of getting down to the mechanistic level, but still being able to look at large scales. And then you have to ask yourself questions such as how deep down do you want to go? Which level do you want to look at in these mechanisms? And that's also an exploration that needs to take place. All of those depend on having the right tools, being able to look at the system. Whenever you want to turn something into a scientific question, when you really want to talk about something like consciousness as a scientific problem, then you need to be able to apply the scientific method, which means you need to be able to build very clear hypotheses, describe what it is you're looking for, what you're assuming or not assuming, and then be able to carry out some experiment where you make measurements, and then those can either prove or disprove. Well, usually you can only disprove your hypothesis. You can find something wrong with it. And that way you can iteratively find your way towards the truth. So this is really what has to happen. More measurement and more working with specific hypotheses. This is exactly what you do in the exact sciences. I'm using the word exact too much. Um,
It's a good
and
word.
in computational neuroscience, where that's the way you go about things. You develop hypotheses about specific circuits and about how they operate, how neurons in there are communicating with one another, what information they're processing, which signals are important, and how they produce their output, what that output then means. All of those things need to be constrained by good hypotheses and by good measurements. So I think those are the big questions, and that's how you should tackle them, probably.
Excellent. Well, yeah, I'm very much looking forward to going to the GF2045. Um, there's one more question that we've touched on before that I thought I might ask you, and that's the brain-body problem. And this is often brought up by philosophers in general, but I think it's important as well. And you've mentioned its importance, so I thought I might conclude by asking you what you think is important in tracking the embodiment problems. I mean, should we just
Yeah,
upload? Should we just consider just how the brain works, or how the body works in conjunction with the
right.
brain?
Yeah.
Should we be simulating the body and the brain?
I would love to talk about embodiment, but before we jump into that,
Hmm.
because you already used the word uploading, maybe I should quickly say
Okay.
what that often meant to be.
Yes, yeah.
Um, so when we talk about the interesting experiences of what's going on inside us,
Mm.
what being really is, we're talking about mental processes. We're talking about the sorts of functions that the mind is running, and they're running on the mechanisms that are the biological brain. Now, those functions that are being carried out by biological mechanisms, they could also, in principle, be carried out by anything else that can produce the same type of output for the same sort of input. When you do that, when you run it in some other type of implementation, then it's no longer a biological brain, it's another brain, but it could have the same mental functions. Then we say that our mental functions have become substrate independent. So we call this substrate independent minds, or SIM. When you move from biology to another implementation, we call that mind uploading. This transfer from the biological implementation to an implementation in another substrate. And one of the ways of going about this is to replicate what each of those components that are important inside the brain, such as neurons and synapses, are doing. And when you replicate that, that's what we call whole brain emulation, or just brain emulation in some cases when we're talking about pieces of the system. So, you were asking about the embodiment question, which is okay, so let's say that we are emulating what's going on in the brain. How important then is the rest of the body? How important is to our experience, our being, this set of mental functions? How important is the rest of what's out there? What does this mean? And I think it's pretty well agreed upon, or at least it's agreed upon in neuroscience and in some areas of artificial intelligence, that embodiment is very important because our experience or sense of being isn't just what's being computed in the brain. It is, of course, that which is being delivered to the brain and then being processed, and how our output affects the external system, and then again, what that causes us to experience. So it's a constant feedback, it's a loop feeding back from our environment to us. And the way that we experience the environment is through our senses, which is again through our body and through our actuators, which are in our body. That's how our output can affect the environment and then again change this feedback. So, embodiment in an environment is 100% relevant to the experience of being. And I think that by putting both the body and brain questions into one Congress, we're we're accentuating that. We're emphasizing that indeed we understand that this is one and the same problem. We can't just deal with one and not the other. And we do have to deal with both, especially if we're talking about something that we want to apply as a possible set of solutions to problems that humanity is going to face, challenges that are coming on, which are, of course, us existing within our environment, the entire experience of being, of the human experience, in a sense.
Yeah, excellent. All right, well, very much looking forward to it. Is there anything else that you'd like to cover off about the conference that people should know? Like,
No, not right now. I think we've pretty much said it all. The conference is the Global Future Congress 2045. You can go to the website at gf2045.com, where you can see everything about participating and join all of us who have been discussing these wonderful topics there for 2 very interesting days. We're finally we'll put all the pieces of the roadmap together, talk to some of the speakers who are doing the cutting edge work in those areas, and just see that we can move this on to even greater things as the convergence of interest in brain sciences seems to be happening just now.
Yeah, it's the right time for all these things to happen. I mean, the technological infrastructure that we have, and now that we can communicate very easily through the internet, and that seems to be increasing the efficacy and speed of research, which is quite amazing. I don't think a lot of people really realize how unique this sort of time period is in our history. It seems like it's a very strange but wonderful and exciting time to be
Mm,
alive, and just to be able to see some of there's certain technologies which are going to have a massive impact, just like the brain. Our human brain, our ability to
yeah.
abstract and conceptualize had a massive impact on the world, on the rise to prominence within the animal kingdom. The
Mhm.
study of neuroscience, the study of the brain, is going to have a really large impact on the state of play in our future. We are heading towards a very exciting time and it's exciting time to be alive. It's going to get more exciting in the future, especially if we focus on this sorts of research. I believe it's one of those areas which have probably the largest headroom
Yeah.
for future development and possibility in the future. So yeah, I'm excited about these sorts of conferences. This GF 2045 seems to be connecting a lot of the right people together. So yeah, I encourage as many people who can make it to witness this convergence, as you said, of brilliant minds and brilliant technologies together. So yeah, come along if you can. Yeah, I certainly will do the best I can to get there. I'm in Australia, and New York is half a world away, but it'll be a wonderful time. So yeah, all right.
Yeah.
Well,
I'm sure
cheers.
you'll make it over there, and I look forward to seeing you at Congress.
Yeah, if I can get time off work, but yeah, for sure. All right. Well, thanks so much for your time, Randall, and looking forward to chatting with you again. So signing out. Cheers. Bye then.